Hello everyone, in this video I'll be making notes for the process of transcription, the first stage in protein synthesis. All of this process takes place in the nucleus. We start with a molecule of DNA, a section of that DNA we call a gene. The section of DNA is unzipped. This is catalyzed by the enzyme RNA polymerase, which binds at the start of the gene. This exposes one template strand. RNA polymerase will have a second role later on in this process, but its initial role is to catalyze the unzipping of the gene, unlike in DNA replication, where DNA helicase is used for this process. In the nucleus, there will be 3 RNA nucleotides. Each RNA nucleotide is attracted to its complementary base. We call that process complementary base pairing. You must remember that thymine is replaced by uracil whenever we are making any form of RNA molecule. A pairs with U for uracil, G pairs with C, C pairs with G, and T pairs with A. Once the nucleotides have paired up, we still need to join the nucleotides together and form the sugar phosphate backbone. This is also catalyzed by RNA polymerase. This enzyme catalyzes the condensation reaction to form the phosphodiester bonds. This makes the sugar phosphate backbone. So our RNA polymerase has had 2 roles: binding at the start of the gene to unzip and catalyzing the formation of the phosphodiester bonds to join the nucleotides. Once the nucleotides have been joined, we have a molecule called primary mRNA, also called pre-mRNA. This molecule is composed of introns and exons. Introns are sections of DNA that do not code for a sequence of amino acids, whereas exons do code for a sequence of amino acids. We need to remove these introns as they don't code. This process we call splicing. Once the introns have been removed, we are now left with a shorter mRNA molecule that is composed only of exons. This is our mature mRNA, often just called mRNA. mRNA is able to leave the nucleus via the nuclear pore, so that the next stage of protein synthesis can take place. The mRNA is a complementary copy of the genetic code. DNA is unable to leave the nucleus. However, mRNA can. So this molecule carries the genetic information so that translation can occur. Watch my video about translation so that you can get detailed notes to aid your revision. Subscribe to my channel and do drop me a comment if there are any videos you'd like me to make.